All right, let's go ahead and get started. Good morning. Welcome to the Harmful Algal Bloom webinar series. My name is Amy Weckel, and I'm from the Illinois Water Resources Center, located at the University of Illinois in Urbana-Champaign, and I'll be your moderator today. I apologize for the train that just decided to go by. This webinar is hosted by the Algal Bloom Action Team, of which the Illinois Water Resources Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resource Research Institutes, the North Central Region Water Network, and university extensions within each state in the North Central region. In addition to hosting a webinar series, our team is developing a website of resources including fact sheets and frequently asked questions on HABs topics. I encourage each of you to visit our website, which I'm putting in the chat now. There you can explore some of the resources we have to date, including a What You Should Know fact sheet, a HABs frequently asked questions database, and recordings from previous webinars, and our virtual HABs Research Symposium, which was held in January. Drop that website. Before we get started, just a few housekeeping items. We will have a Q&A session after our speakers present today. Please list your questions for our speakers in the Q&A panel, and we will do our best to address all of them following the presentations. If there is a question already posted to the Q&A panel and you have the same question, upvote the question and we'll be sure to get to the answers to the most popular questions during the session. Today's webinar is being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you are having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well. We will be happy to assist you as best we can. Today's presentations focus on the removal of cyanobacterial blooms, and we have two great speakers lined up for you. Our first speaker, Dr. Halis Simsek, is a faculty member in the Department of Agriculture and Biological Engineering at Purdue University in Indiana. He has been active in algal research for the past 20 years. His team mainly explores the application of algal cultures for wastewater treatment. Currently, he is working on cyanophages and algal viruses for the removal of harmful algal blooms from the environment. In addition, Dr. Simsek also explores the downstream processing of algal biomass for the production of value-added products. Dr. Simsek has secured grants and published articles on algal and cyanobacterial cultures and their implications in wastewater systems. Following Halis's presentation, we will hear from our second speaker, Dr. Pankaj. I'm sorry, massacred that. Pankaj Bhatt, and he is working as a researcher at Purdue University in the Department of Agriculture and Biological Engineering. He did his undergraduate in biotechnology in 2009 and postgraduate in microbiology from Kumaon University in India. Furthermore, he completed his doctoral degree in microbiology in 2015 from G.B. Pant University in Technology in India. In the past 10 years, he has been working on various aspects of environmental biology. Currently, he is investigating the impact of harmful algal blooms on the environment, and he is actively involved in the isolation and characterization of cyanophages from various water samples in the USA. He is cultivating the algae using synthetic media as well as with wastewater. In addition, Dr. Bhatt is working on the cyanobacterial cultivation and isolation of cyanophages under different conditions. This study will explore and give insight into pictures of the cyanophages and their interaction mechanism with cyanobacteria. Once again, don't forget to put your questions in the Q&A panel and we'll address them following the presentation. And I think we're ready now, Anne, if you would like to, I will stop sharing.
All right, great. Oh. All right, we're going to start with Dr. Simsek, and so let me know if you're not able to hear the presentation.
Hello everyone. Today I would like to talk about Cyanophage for removal of toxic blooms. My name is Halis Simsek. I am an assistant professor at Agricultural and Biological Engineering Department at Purdue University. Outline. I will give some information about cyanobacteria and removal of cyanotoxins and biological removal of cyanobacteria. Then I will talk about cyanophage and interaction between cyanophage and cyanobacteria. As we know, we can use cyanophage to control cyanobacteria, but I would like to present how we can control cyanobacteria in the lake environment, which is a real environment. Are aerobic.
Sorry about that.
Cyanobacteria, also known as blue-green algae, are aerobic photoautotrophic prokaryotes, and they are able to reduce molecular nitrogen to ammonia in water environment. Excessive proliferation of predominant cyanobacteria genera in water bodies usually forms surface blooms. And certain types of cyanobacterial blooms can produce cyanotoxins that are harmful to the environment. Most cyanobacteria produce phycocyanin, which gives cells and certain types of cyanobacterial blooms can produce cyanotoxins that are harmful to the environment. Most cyanobacteria produce phycocyanin, which gives cells a blue color that can be visible in higher concentrations. Overgrowth of cyanobacteria in a water environment is not a simple process that is caused by a single factor, but rather multiple factors occur simultaneously. Excessive nutrient loading through treated or untreated sewage systems, agricultural runoff, or industrial discharge cause the proliferation of cyanobacteria in the water environment. In addition, warm water temperature and the composition and characteristics of the water ecosystem will be opportunities for the cyanobacteria to grow. This picture shows before and after contamination, cyanobacteria contamination. As you see on the right picture, cyanobacteria covered all the water surface. Cyanobacterial harmful algal blooms, which also called cyanoHABs, are increasing throughout the world and contaminating aquatic ecosystems. Poor watershed management and various environmental conditions increase the HAB occurrence in the surface waters. Phosphorus accumulated at the bottom of the lake could be released during the summer and promote cyanobacterial growth. When anaerobic conditions started underneath the lake, then phosphorus could be released. The blooms cause severe environmental problems, including unpleasant taste and odors and cyanotoxin productions. Cyanotoxins have been reported to be lethal agent for living beings that treat animals, people, and aquatic species, drinking water reservoirs, and recreational condition of surface waters. These are some type of cyanobacteria, and these are common cyanotoxins. Microsystem, Cylindrospermopsin, Saxitoxin, and Enotoxin A. These metabolites must be removed when they contaminate the drinking water sources. Cyanobacterial population growth and surface bloom formation in marine and freshwater environments have been elucidated by researchers. However, the conditions that cause the decline in cyanobacterial population in water body have not been addressed properly. Viral infection could be one of the reasons for the population decline. For example, a study proved that the populations of Cyanobacteria and Cyanophage are positively correlated. A decline in Cyanophage causes an increase in Microcystis aeruginosa. Cyanohabs in the lakes are another important topics that I would like to bring. For determining the best management practices to suppress cyanohabs in the lake ecosystem, we need to know if the lake serves as a reservoir for an adjacent drinking water treatment plant, if the lake receives any upstream discharge from any wastewater treatment facility, if any river or stream is introduced continuously to the lake. And also we want to know if any water is withdrawn from the lake by humans for some purposes. If the recreational activities on the lake are allowed, then if the lake regularly experiences cyanobacterial blooms that cause taste, odor, and turbidity in the lake. And finally, the copper sulfate or other algicides has been applied to the lake to stop cyanobacterial growth. We need to know all these items. Removal of cyanotoxin. Effective treatment options should be applied to remove cyanotoxins from the drinking water. Conventional drinking water treatment methods are not effective to remove cyanotoxins. Chemical and biological treatment options for cyanobacteria should be investigated. However, the chemical treatment might produce harmful byproducts in the system. During the drinking water treatment process, removing the cyanobacteria in the disinfection unit or using copper sulfate may create stress conditions on the cyanobacteria and cause to produce cyanotoxin in the water. Therefore, even though the cyanobacteria are removed from the water, the toxins may remain in the water. Powdered activated carbon or granular activated carbon can be used as a pre-treatment method to remove toxins. Nanofiltration or reverse osmosis are both used effectively to remove cyanotoxins from drinking water sources. Now I would like to talk about biological removal of cyanobacteria. Cyanotoxins should be degraded biologically in the water ecosystem, which is a good thing. Implementation of biological filtration is one of the promising methods to degrade cyanobacterial metabolites. The water contact time and hydraulic retention time are crucial for the biodegradation of metabolites. Most of the cyanobacterial metabolites present in the water bodies are susceptible to biodegradation by bacteria. Studies showed that microcystin in drinking water sources, wastewater lagoons, and stabilization ponds can be degraded by bacteria. The biodegradation of microcystin doesn't produce toxic byproducts. Cylindrospermopsin can be degraded by bacteria as well in a natural water ecosystem when the temperature are appropriate and at the presence of copper-based algicide. Biodegradation of saxitoxin and anotoxin A by bacteria in the water environment also has been documented. So overall, we can say that 4 main types of toxins can be removed biologically. Now I would like to talk about cyanophage. Viruses are the most widespread and abundant biological entities in the environment. Bacteriophage, also we can say phage, is a virus that infects bacteria. Bacteria will be host. Cyanophage is also a virus that infects cyanobacteria. In this case, cyanobacteria will be host. Cyanophage technology helps to protect the environment by controlling the cyanoHABs in water ecosystems. In addition, the technology can be used for rapid detection of cyanobacteria in the aquatic system. Now I would like to talk about cyanobacteria and cyanophage interaction because this interaction is very important. Cyanophage must infect cyanobacteria when the perfect conditions exist. Cyanobacteria and cyanophage represents a diverse group in marine and freshwater environments. They both can live in every condition and can adapt to new conditions. Successful replication of cyanophage in the host depends on the environmental conditions, including temperature, rainfall, flood pulse, turbidity. In practice, there are some biotic and abiotic factors that influence cyanophage infection, lytic and lysogenic, in a natural microbial community, including rapid increase of host mutant in the water body, phage-resistant mutant. So we want phage infect cyanobacteria, but there are some problems. Another problem is the changes in the morphology of the cyanobacterial cells that prevent phage interaction. And water temperature and water column, solar radiation, pH, cation concentration, and salinity, seasonal variation, microbial communities, all these parameters are important. I would like to give some information from the literature about cyanobacteria and cyanophage interaction. One study was conducted to investigate cyanophage infection and lysis of filamentous cyanobacteria. The samples were collected from the coastal water in Sweden. Then they investigated removal of hepatotoxin modularin. The experiments were conducted in the laboratory condition. They found that the cyanobacteria cell density was reduced by 93%. So this study shows that cyanophage can destroy cyanobacteria very well. Another study reported a cyanophage infection study on the marine cyanobacteria under light-dark conditions, and they monitored the phage replication. They found that phage was produced in the light but was not produced in the dark. Another study proved that a cyanophage successfully infected and took over the host metabolism of marine cyanobacteria. Then one more study compared to the removal of Pseudomonas aeruginosa biofilm in wastewaters using chlorine and bacteriophage. So this is bacteriophage, not cyanophage. So we are talking about bacteria, not cyanobacteria. in this slide, they found that chlorine removed 40%, whereas bacteriophage removed 89% of bacteria. They also found that consecutive application of bacteriophage and chlorine removed 96% of the bacteria. That means first they applied bacteriophage, they removed some, later applied chlorine, then total they removed 96%. From here we can connect this study to the cyanobacteria. We can conclude that consecutive applications of cyanophage and chlorine could be applied to remove cyanobacteria in a drinking water treatment plant. I couldn't see any study on this. Maybe available. Maybe it is available. I am not sure. Another study reported that cyanobacteria play an important role in the photosynthesis and carbon dioxide fixation process in the environment. They conducted experiments to address the question of how cyanophage infection could alter the photosynthesis and carbon dioxide fixation during the infection. They isolated cyanophage and a cyanobacterium from a lagoon. They found that photosynthesis and carbon dioxide fixation activities were reduced in the host because of viral infection. Overall, cyanophage-cyanobacteria interactions are well studied in laboratory conditions, and the following parameters were measured and monitored: phosphate and nitrate uptake, photosynthesis and carbon dioxide fixation, the impact of light-dark cycle, solar UV radiation, composition and characterization of cyanobacteria, and characteristics of one environment. If the replication speed of cyanobacteria is faster than infection of phage, then cyanohab can occur. Other environmental conditions are also important. There are several reproduction cycles of cyanophage. Lytic and lysogenic cycles are the most dominant cycles. Previous studies showed that nutrient availability and other environmental conditions may cause cyanophage to switch the reproduction cycle between lytic and lysogenic in water environment. So why this is important? We have to monitor cyanophage reaction in lytic condition and lysogenic condition. Whichever condition is best for us for certain lake, certain environment, then we may force bacteriophage to work in that cycle. I would like to talk about Cyanophage application. Cyanophage can be found in the same environments inhabited by host cyanobacteria, and each cyanophage will target only specific bacteria. Phage in a lake environment should be able to adapt to various environmental conditions and survive against environmental and external forces. So that means in the laboratory we have controlled environment. However, in the real lake, situation will be different. And phage should survive in that real environmental conditions. Cyanophage efficiently replicate within the host cell and ability to stay dormant in the host. Of course, if environmental conditions are met. Now, I would like to continue about the cyanophage application on the lakes. To prevent cyanohabs occurrence in the lake and reservoirs using cyanophage technology, the parameters that need to be considered are as follows. water circulation rate, depth of water, seasonal temperature variation, heat transfer, nutrient loading, sunlight exposure time and intensity, wind effect, stagnant water, circulation, turbulence of water, volumetric flow rate, residence time are important. Now I would like to conclude my presentation. The relationship between cyanobacteria and cyanophage can be managed by adjusting some environmental parameters in the lake. In other words, on one hand, an excess nutrient loading in the lake will promote the rapid growth of cyanobacteria, causing the bloom. On the other hand, the type and concentration of nutrients can support the efficacy of the cyanophage infection in the cyanobacteria, thus increasing the cyanobacterial lysis. This natural biocontrol cycle should reduce cyanobacterial production in the lake environment. Laboratory experiments are helping to understand the phage-host relationship. However, the application of cyanophage to control cyanobacteria in a surface water body is still challenging. Cyanophage have the potential to improve the quality of surface waters if we can understand the interaction of cyanophage cyanobacteria very well. Prevention, control, and mitigation of cyano-HABs in surface waters will be inexpensive, rapid, and environmentally friendly. Mixed culture cyanophage cocktail could be formulated to terminate all the cyanobacteria species in the lake environment. However, cyanophage may not kill all the host cells, and phage-resistant host mutant may reform the bloom. Then high concentration of toxin release may occur and spread in the lake. So in one side we can kill all the cyanobacteria, but in other side cyanobacteria will be under stress condition, then release cyanotoxins. We will clean cyanobacteria, but toxin will remain in the water. As a future study, I can advise that cyanobacteria and green algae relationship should be studied, and there are many studies are available also. In the lake environment, which organisms will outperform, cyanobacteria or green algae? If these two different types of algae are together, which one will be dominant? Some studies explained that the green algae predominated over cyanobacteria, while other researchers explained excessive nutrient entrance to a lake environment enables cyanobacteria to suppress algal growth. If nutrient loading is high in a lake, we should monitor if cyanobacteria will be outperform on green algae. Then this can be an advantage. We can use this information to manage the lake. Therefore, the impact of nitrogen and phosphorus ratio on algal and cyanobacterial growth in the lake environment must be studied. These are my references. And thank you.
All right, great. Thank you. Sorry about that. Thanks for bearing with us as we were figuring out how to do this here. I'm going to go ahead and share the next one. I'll try to make it a little bigger for folks. Again, let me know if it's not working.
Hello everyone, I am Pankaj Bhatt, working as a postdoc in Agriculture and Biological Engineering Department of Purdue University. So here I am giving my presentation on Cyanophage cyanobacterial interactions and its application in cyanobacterial blooms removal. So here I am going to talk about cyanobacterial blooms formation and growth curve, cyanophages structure and classification, host range specificity of cyanophage, cyanophage cyanobacterial interactions, cyanophage life cycle, and genomic and metabolic activities of cyanophage in cyanobacteria. If we see the brief about the cyanobacteria bloom formation, so then we can see here the blooms are formed by several ways, and there are many regions. So the blooms can be formed by the industrial fossil fuel emissions that creates the harmful gases that deplete the ozone layer, which ultimately creates the global warming and increase the temperature of the water. In addition to this, the nutrient loading from the different human activities also increasing into the water environment. So urbanization, agriculture, residential and agriculture waste disposal creates organic and inorganic nitrogen and phosphorus, carbon continuously into the water environment, which leads to the changes in the water environment for the eutrophication, as well as there are much blooms are formed that creates the toxicity in water environments. So although all cyanobacterial blooms are not toxic, but some of the strains are toxic because they can produce the specific type of metabolites in water system as a bloom, so that are anoxious for the growth of the other living being inside the aquatic environment, as well as it can reduce the living things possibility in water. Here we are talking here, this is the growth curve of the cyanobacteria. So because cyanobacteria is a bacteria, so it can grow with 4 different phases, that is called lag phase, where it adapts in the initial environment for the nutrition. Then it enters into the log phase, where the continuous growth of the micro cystis like and other cyanobacteria, which can grow exponentially in the log phase. It is also called exponential phase. So in exponential phase, these bacteria produce the metabolites. These metabolites are cyanotoxins in nature. So there are different types of cyanotoxins, as already described by Dr. Simsek in previous presentation. So here I am talking in brief. So microsystems are toxic, and the maximum synthesis of microsystems in environment during log phase of the growth, whereas minimum production was achieved in stationary phase of the growth. So in this way we can understand the growth curve linked production of the cyanotoxins. Here now cyanophages. So cyanophages are a type of bacteriophages. So they can eat the bacteria. So here cyanophage eating, these bacteriophage eating the cyanobacteria. So that's why we are calling them cyanophages. So they are present in both marine and freshwater environments because their hosts are present in both the environments. So Cyanophages contains double-stranded DNA as a nucleic acid. Cyanophages are responsible for 10 to 80% of cyanobacterial mortality in natural environment. If we see the structure of cyanophage, so we can see there is a capsid head, nucleic acid, collar, sheath, base plate, spikes, and the tail fibers. So there are different types of cyanophages existed in environment. So many of them are already reported, but still there is needs to develop more technologies which can isolate and identify more cyanophages from the water environment. So here is the example where we can see the cyanophage min S1 infecting the Microsystem aeruginosa. So these in the plates we can see the clear plaques of the Microsystem aeruginosa in this study. So cyanophage was first discovered by Sefferman and Morris in 1963. So the first cyanophage was named as LPP1 because it can infect Lyngbya, Planktoniema and Formidium. So to date, there are 12 cyanophages are identified for the infection of in Microsystem species, and some of the cyanophages can infect the broad host range, where a few of them are specific for the single stains. So it depends on cyanophage to cyanophage how it proceed for the infection in different environment. Cyanophages properties matched with the bacteriophages such as T7 phages, but modes of action is different. The host, on the basis of host range, the cyanophage is categorized in different groups, that is called LPP group, A group, AN group, NP group, AS group and SM group. So ecologically they are present in both marine and freshwater environment. The host range and specificity, the LPP group cyanophages mainly it attacks to the three genera of non-heterocystous filamentous cyanobacteria, that is called Lyngbya, Formidium and Planktoniema, whereas A, AN, N and NP groups infect the Anabina, Nostoc and Planktoniema species, and AS and SM it stands for Anacystis and Synechococcus and Microsystem genera of the Cyanobacteria. So here the International Committee of Virology divided the cyanophages into three broad categories. So these categories are Podoviridae, Myoviridae, Cephuviridae. So Podoviridae generally it contains the phages of short bulky tail, and Myoviridae it contains the phages of 160 nanometer tail, non-enveloped, as well as whereas in Cephuviridae there are short long tail without tail fibers and non-enveloped. So in Podoviridae, the categorized viruses are MALBP 5MHI142, MALM01 for Myoviridae, and MAMVDC PLP. These are the different types of viruses that comes under the different families of the cyanophages. So cyanophage makes the special interaction with the cyanobacteria in natural environment, so they can interact with the host bacteria using the receptors in their membrane as well as with the tail fibers of the cyanophages. So there are so many factors responsible for the cyanophage-cyanobacteria interactions, such as depth of the water, temperature, solar radiation, pH, cation concentrations, salinity, seasonal variations. So if we see the life cycle of cyanophages, so there is two types of life cycle present in the cyanophages. One is called lytic cycle and another one is called lysogenic cycle. So in lytic cycle, cyanophages grow themselves, it attack to the host cyanobacteria and it creates its progeny, then it releases the progeny by bursting of that cyanobacterial host. So in this case, cyanobacteria degraded due to the multiplication of the virus. So in lytic cycle there is 4 steps: cyanophage attachment, entry into the cyanobacterial cells, cyanophage replication and production of new cyanophages. Whereas in lysogenic cycle, also known as temperate or non-virulent infection, so in this cycle there is no infection caused because this cycle is mainly for the replication and the cell division of the cyanobacteria and the replication of cyanophage genome. So there is no host lysis. So whereas in injection of cyanophage DNA into the cyanobacterial genome, so prophage formation. So prophage is a structure in which the cyanophage DNA integrates with the cyanobacterial DNA. So in this cell division and derived replications observed, and transition from lysogenic to lytic occurred due to stress such as UV light, low nutrient and mutagen. And the transition from lytic to lysogenic is called induction. So here we can see that the cyanophage life cycle in lytic cycle, phage attaches to the host and it creates its progeny, and the host cell is burst and it degraded. So here in this presentation, as we are interested in removal of harmful blooms, so the lytic cycle is important, but in addition to lytic cycle, lysogenic cycle is also very important. So in lysogenic cycle, phage DNA integrates into the host chromosome of cyanobacteria and it replicates its DNA, as well as there it creates the cell division and form the new cells which contains the cyanophage DNA. So next again it enters into the lytic cycle and again it releases the new. So now isolation and identification of cyanophages. So the question is that how we can isolate the cyanophages from the environment. So for that we can collect the water samples from the regions where the cyanobacteria blooms is formed or where cyanobacteria may exist. So such because this bacteria cyanophages interact with its host mainly, so the sample collection site is very important. Then we can preserve the samples at 4 degree until use, or we need to do the plaque assay as soon as possible. So in addition to the cyanophages, we need to also grow the cyanobacteria like Microsystem aeruginosa is an example. So we need to grow the pure culture, then we need to go for the plaque assay. For plaque assay, we just prepared the top agar plus active cyanobacterial culture. So in that we are using 100 microliter of active cyanobacterial culture plus 100 microliter of the cyanophages as a water sample. So and then we poured together in plate. We after 6, 7, 8 days we can find the clear plaques that on BG11 plates, BG11 agar plates of cyanobacteria with cyanophages. So here, in addition to cyanophage, here we can also see that bacteriophages infecting bacteria also makes the clear zones called plaques. So cyanophage plaques and bacteria plaques, they are clear like a clear zones, and there is no much difference than the host bacteria. So in this case, the cyanobacteria plaques and the bacteria plaques are different on the basis of their morphology and culture growth conditions. So in this slide I'm explaining about the 5MA05 impact on the removal of microcystic blooms. So here we can see that the Necken et al. Reported the cyanophage 5MA05 which killed the microcystis in different days, and after 13 days the growth of the microcystis completely disappeared and the production of cyanophage was maximum. So this the TAM and transmission electron microscopy structure shows that cyanophages are effectively infecting the microcystis species, and it lyses the cells very. So in this slide we are discussing
again.
quickly. So now abiotic and biotic factors regulating the cyanophage cyanobacteria interactions. So in this slide we are describing how cyanobacteria blooms formed and how they are linked with the other factors. So the biotic factors is that there is the cyanobacteria that is killed by the lytic cyanophages, which ultimately convert, which ultimately reduce the harmful algal blooms. Whereas due to the rich phosphorus, it grow in large quantity. So there, so this is the slide representing the cyanobacteria and in bloom formation and the toxicity of cyanobacteria in environment. So ultimately it produces the cyanotoxins. The solution is that the cyanophages can infect the cyanobacteria blooms as a cell, and it can remove the blooms very clearly, and it can clean the waters and makes the environment sustainable for the living beings. So now the cyanophage genome. So all the actions of the cyanophages is mediated by the genomic sequences, because it contains the diverse genomic sequences inside the capsid. But for the molecular metabolism, it depends on the host cyanobacteria. So complete genome sequences show that the unique genetic resources in cyanophages. Cyanophages acquire diverse genes from host genome, and host-derived genes contribute to the ecological success of cyanophages, because if cyanophage contains the host genes, it can express the genes according to its fitness. So genes encode the specific proteins and enzymes are beneficial for cyanophage life. Comparative genomics suggested the majority of the genes showed similarity with the host DNA sequence. Cyanophages infect Prochlorococcus and Synechococcus carry the PSB-A and PSB-D genes, which play a role in photosynthesis. So this is a very important finding by previous researchers. The two genes PSB-A and PSB-D that are very important for the photosynthesis in cyanobacteria, it can be coded by the cyanophages. So it means that cyanophages is taking the advantage of host machinery for its own benefit. If it wants to survive up to a long time, it can produce more D1 and D2 protein. So host can take the advantage of photosynthesis, and cyanophage can take the advantage for its nutrition energy from the host cells. The origin of host-derived metabolic genes. So the cyanophages contain the genes that is already present in host, but after horizontal gene transfer, it comes from the parent cyanophages to progeny cyanophages. Consequently, the PSB-A gene was observed in more than 50% of cyanophages infecting Synechococcus in the Red Sea. The photosystem's second core reaction system encoded PSB-A 80% cytomophages, whereas 50% of the cyanophages encode both PSB-A and PSB-D genes. Acquisition of photosynthetic genes in cyanophages is a wider event, so it is a complex relationship. Freshwater cyanophages encodes for the NBL-A gene that degrades the phycobilosomes in cyanobacteria. Host-derived genes help cyanophages for ecological fitness. Cyanophages encoded host genes affect the central metabolic path in host cyanobacteria. So it means that host-derived metabolic genes are beneficial for cyanobacterial life. Here we can see that when cyanobacteria infect the host, its DNA come inside the cyanobacterial cells. The genome of the cyanophages is transcribed inside the cyanobacteria. It produces different enzymes and proteins that ultimately affect the photosystem second, which is able to the transfer of sunlight to the biological energy using the electron transport chain. So in this figure we can see that the cyanophage host-derived genes ultimately affect the cyanophage metabolism as well as production of new cyanophages. So if cyanophage produce these genes, it can produce more progeny inside the host cells. So in another study, we found that some of the authors collected the water sample from the Lake Erie, and they isolated and screened the potential cyanophages that could degrade the Cyanobacteria Microsystem aeruginosa. So this is they given the name Ma, and Ma they are giving for Microsystem aeruginosa, and LEP infecting the Microsystem aeruginosa. Ma is the strain of Microsystem aeruginosa. So MALEP affected the M. aeruginosa with dynamic changes in chlorophyll and phycocyanin. So cyanophage MALEP negatively affect the photosynthesis, and the mechanical stiffness of M. aeruginosa membrane observed after cyanophage infection. So the conclusion is that of this work is that the cyanophages isolated from the lake have the potential to degrade the Microsystem aeruginosa, and it deformed the membrane, and slowly it completely break down the membrane and converted the blooms into, and tried to helpful for the removal of blooms from the environment. So in conclusion, we can say that the human activities increasing the nutritional load of the wastewater, nutritional load of the water system, which leads to the formation of cyanobacteria harmful blooms. Further needs to understand the complex microbial communities in the water environment. Cyanophages are able to treat the cyanobacterial harmful blooms, which is a promising technology for sustainable environment. But problem is that the massive cyanophage required for removal of the large blooms. So we need to figure out how we can apply the technology. So the future research directions can be on the basis of these research work. The future research direction could be the behavior of cyanophages and the cyanobacteria involved in cyanobacteria harmful algal blooms, interaction between cyanobacteria and the cyanophages. So we must know how Cyanophage will attach to the cyanobacteria. So if we solve this puzzle in more detail, so we can effectively try for the algal blooms removal. In addition to this, we also need to know the physical characteristics of the cyanophage interactions, identification of novel cyanophages, and development of molecular markers. So the developed molecular marker could be helpful for the isolation of the cyanophage community from the big lakes. So for that, some people are already using the qPCR. So some markers are identified, but still there is lots of work we need to do for the identification of the cyanophage quickly. So overall, these research directions could explore more about the cyanobacteria interaction that could be helpful for the removal of cyanobacterial blooms completely from the environment. So these are the we use. So thank you very much. So I want to say thanks to my supervisor, Dr. Halis Simsek, for giving me the opportunity to learn this interesting cyanophage work. So really I am enjoying this work, and I am expecting for the more results in future. Right now we are in starting phase. We are doing the plaque assay for cyanophages, and maybe in future we will have more interesting results that we would like to share you again in the coming webinars. So thank you very much. If you have any question, you are welcome.
Thank you so much. Those were really nice presentations. I'm going to go ahead and share my screen again. Okay, I'd like to thank everyone for participating in the Algal Bloom Action Team's webinar series today. A special thanks to our presenters, Halis and Pankaj, I'm so sorry, for sharing their research with us. And also thank you to Ann Nardi and her team at the North Central Region Water Network for hosting the Zoom webinar. in coordinating the technical aspects of the webinar. We couldn't do it without you. As we move into the Q&A session of the webinar, I'd like to remind you to check out the Algal Bloom Action Team's website, which I'm dropping in the chat again. Here you'll be able to connect with the team and subscribe to our events. Our webinar series will continue bi-monthly throughout 2022. The next one will be on October 5th, again at 11 a.m. And we will now move into the Q&A session. We will draw initial questions from the comments from those submitted in the chat box during the presentations, and we will do our best to get to all of them, though I know we're running short on time. Following the Q&A session, please complete the survey at the link I'm also providing in the chat, and we appreciate any additional comments or feedback you have for this series. Okay, our first question is, if Cyanophage causes mass lysis of cyanobacteria cells, will the toxins degrade quickly enough to prevent effects like fish kills?
Yeah. Yes, can you repeat the question again?
Yes. If Cyanophage causes mass lysis of cyanobacteria cells, will the toxins degrade quickly enough to prevent the effects like fish kills?
Yeah, actually, yeah, this is the one problem. Like if the Cyanophages will degrade the blooms, so toxin will release into the outer environment. So in this case, the surrounding living things will be affected. So for that, we have seen there are some researchers are working, and we are also thinking to work on this. Some of the indigenous aquatic microorganisms that could degrade the toxins in that after releasing into the environments. So I think that would be the solution to prevent the toxin to come and stay into the environment up to a long time. So because otherwise, if we are treating with cyanophages, so again, it will create the water system contaminated with cyanotoxins in large scale. So I think the best way is that to minimize in biological manner, we can think about the degradation of microsystems and other cyanotoxins surrounding the blooms after Cyanophage application.
Thank you. Halis, did you have anything to add?
Yeah, probably the toxins will remain longer and it will continue to affect life in the lake or surface waters. So for Phage, the job for Phage to kill the cyanobacteria, they will not treat, they will not remove the toxin. So toxin will remain in the waters. But during that activity, cyanobacteria will release toxin, as we mentioned, because of stress condition, and that toxin will stay in the water. I don't think it will be removed. It will degrade itself in short time.
Okay, thank you. The next question, and I think this might be our last one. So on the screen, you can see the speaker's contact information, and I believe it's okay. Can participants who didn't get their questions answered reach out to each of you after the seminar? Okay, that would be great. Thank you. The last question will be, what should lake managers do to encourage the growth of Cyanophage to control HABs?
This is not an easy job. It depends on the size of lake, of course, because it is very hard to control the lake because of many environmental conditions. I don't think there are much things available for managers to do, because first the scientists should conduct some research Maybe, I'm not sure if any pilot study available, pilot lake study available or not. I couldn't see any paper. Maybe in a small lake, some research could be conducted, then environmental parameters can be managed. After that, it can apply in big lakes. Before everybody leave, I would like to say one more additional thing. I am an environmental engineer. We are on the engineering side. Pankaj is a microbiologist. So we want to focus on direct application in the lake. As we explained, scientists studied in the laboratory so far. We want to study in the lake. I know that in this webinar group, there are many expert people available. If anybody wants to collaborate with us, please, they can contact us. We want to create pilot scale study in real lake. Could be small size. Just I would like to let people know. Pankaj, do you want to add something?
Yeah, because I have some experience on the degradation of the other synthetic pollutants. So we are also thinking to degrade the cyanotoxins first in lab, how we can treat using algae, bacteria, and fungi in the laboratory. If we will find some best degradation way or the easy way to remove the toxins in the water system, so that will be advantages. So whatever the work we are doing, we want to include all the peoples to exchange our information so we can do more accurately.
And I will add one more thing. Actually, we were planning to present all research data here today, but we couldn't finish all research because we have some back orders, as you know. Whatever we order, it comes late. Because of that, we postponed. I hope we will present all research data as well later. Thank you.
Thank you.
That sounds great. We're looking forward to catching up with you again later. Okay, I think with that, I don't want to hold anybody up. And so thanks again for joining us today, and make sure to reach out to the Algal Bloom Action Team with any additional questions or comments on the webinar series. Thanks so much.
Thank you for the opportunity. Bye.